name is Alexa Finley.
And I'm Shannon Chavez. We're doing our project on Hierro Island's submarine volcanic eruption. Hierro Island, nicknamed Isla del Meridano, the Meridian Island, is the smallest and farthest south and west of the Canary Islands in Spain. It is in the Atlantic Ocean off the coast of Africa with a population of 10,162. A submarine volcanic eruption occurs at constructive margins, subduction zones, and within tectonic plates due to hotspots. It is estimated that approximately 70 to 80% of Earth's magma output takes place here. The first method that scientists used was surveying. 6 cruises were coordinated over the course of eruptive and post-eruptive episodes of the submarine volcanic eruption. From November 2011 to April 2012, 6 surveys were conducted. Biological samplings were performed until late February 2012 to study the effects of the volcano emissions on the pelagic biota, which are animals, plants, and fungi within the region. Epipelagic mesozooplankton and vertical migrant micronectin were evaluated by plankton nest and acoustic sampling. Once the eruption stopped, a second vessel, the RV Cornide de Savdra, performed the 6th and last survey in April 2012. This time, the pelagic trawl, also known as a fishing net, was included to collect larger organisms that had been uncovered by the echo sounders throughout the previous cruises.
The second method used was hydrography. The Seabird 911 Plus CTD measured vertical profiles of conductivity and temperature. CTD stands for conductivity, temperature, and depth. CTD sensors were adjusted as a Seabird laboratory before each use. Water samples were collected using 24 10-liter Niskin bottles. A dissolved oxygen sensor named the Seabird SBE-43 and a fluorometer for chlorophyll, a estimation named the Wet Labs Echo FL, were linked to the CTD unit. Removal of dissolved water and chlorophyll were done to measure the voltage readings of both sensors. The oceanographers profiled temperature and oxygen profiles according to Joint Global Ocean Flux Study, which is the study of the ocean carbon cycle. The temperature and oxygen profiles of each survey were averaged from available stations within the volcano-affected area.
The third method is satellite data. 1 kilometer spatial resolution data around Hierro Island were used to monitor and visualize the progression of the volcanic emissions. Data was obtained from the MODIS Aqua sensor, moderate resolution imaging spectrodiometer, which cleans and clears images. The standard product sea surface reflectance at 667 nautical miles was selected as a tracer of the volcanic plume since this spectral band had been found to be very effective for detecting murky anomalies in oceanic waters. Remote sensing of chlorophyll A was used as a substitute for phytoplankton clustering, but only during the post-eruptive phase as this algorithm is based on the blue-green band ratio, which is also sensitive to non-living particles, including suspended volcanic material. Due to cloudy weather, there are some missing data from the satellite images. To overcome this lack of data, the daily images were arranged with both the previous and the next day's images. After that, remaining missing data was calculated by linear interpolation of the border pixels. Processing of satellite imagery was performed with CDAS, software, which is comprehensive image analysis package for the processing, display, analysis, and quality control of ocean color data.
The fourth method used was acoustic sampling. A hull-mounted Submarine EK60 echo sounder, 7-degree beam width, operating at 38 and 200 kHz, were used for recording acoustic data detection ranges of about 200 and 1,000 meters depth. The arrangement was set at 1024 pulse duration and a 1s negative ping rate. The acoustic data was not available for the upper 15 meters due to the depth of the transducers, 8 meters on the RV Ramon Margalef and 5 meters on RV Cornine de Saavedra. The data was shown to 7 meters below the transducers, which were excluded due to vessel-caused bubbles and near-field effects.
The fifth method used was net sampling. Samples were immediately fixed in 4% protected formaldehyde. In the laboratory, samples were computerized using a scanner in which the organisms were automatically counted, measured, and classified using Zool image software. Taxonomic groups were founded by a manually entered trained setting, achieving a global error rate of 4.7% in the classification. Vertical migrant micronectid were captured using a mid-water fishing net with a 300 square meter area and about 45 meters in length. The netting size was 80 centimeters near the opening, decreasing to 1 centimeter in the cod end. In the last survey, 2 methods were performed indirectly in the migrant scattering layer. The net speed varied between 2 and 3 knots, and the effective fishing time was 1 hour. Samples were fixed in 4% protected formaldehyde for later identification and calculation.
The purpose of this study was to document the change in biota from onset of the volcano to the post-eruptive phase. The oceanographers reported different changes among phytoplankton groups, and the first observation of changes in the vertical distribution of the biota forming acoustic scattering layers. The chemical changes in the water could have created bloom and killed many organisms. The oceanographers also focused on the effects of the volcano on the deep and the migrant scattering layers. All of this could have resulted in significant increase in global warming.
In conclusion, researchers found no significantly different data. The chemical balance of the ocean remained the same and no bloom was formed. Biota is resilient to the submarine volcanic eruption. Figure 1. Position of El Hierro Island and sampling points. The yellow star marks the position of the volcano. Symbols and arrows indicate oceanographic stations and locations of nets. Colors refer to different surveys performed during the sampling period. Solid and dashed frames delimit experimental, volcano-affected, and controlled non-affected zones, where satellite and acoustic data were collected along time series. Figure 2. Sea surface reflectance, SSR, from satellite imagery. SSR indicates the degree of water turbidity and reveals the distribution of volcanic emissions. Red dots are CTD stations, and black lines depict acoustic transects performed between them. Echograms corresponding to the transect lines and oxygen profiles from each station are shown in Figure 3 and 4. Figure 3. The 38 kHz echogram and sea surface reflectance on November 7, 2011. The color scale refers to backscattering strength. Migrant and deep scattering layers, MSL and DSL, as well as both diel migrations are indicated. Acoustic anomalies are also indicated. The MSL weakening yellow triangles and the elevations of the DSL red triangles. Red lines depict the dissolved oxygen profiles established during the acoustic track. The black color in the time scale refers to nighttime and white to daytime.
Figure 4. 38 kHz echogram and sea surface reflectance on the 18th of November 2011. The color scales refer to the backscattering strength, migrant and deep scattering layers, as well as the downward diel migration are indicated. Acoustic anomalies are also indicated. The MSL weakening yellow triangles and the elevation of the DSL red triangles. Red lines depict the dissolved oxygen profiles established during the acoustic track. The black color in time scales refers to nighttime and the white to daytime.
Figure 5. DSL depth throughout the volcanic plume. Horizontal interpolation of the upper DSL depth. Dots indicate the positions of the original data points. The black ones refer to those collected during nighttime and the white ones during daytime. The solid black line depicts the outer contour of the volcanic plume. Relationships between the upper DSL depth and the sea surface reflectance. Black and gray colors refer to data collected during night and daytime, respectively. Solid lines depict logarithmic regression curves fitted to data points. The vertical dashed line refers to the average SSR outside the volcanic plume.
Figure 6. Temperature and dissolved oxygen profiles during the sampling period. The black lines indicate the first survey at the beginning of the eruption. The remaining surveys are indicated by progressively lighter gray lines, post-eruption, in chronological order. Profiles are averaged from CTD cast performed within the affected zone.
Figure 7. Time series with average parameters collected within the volcano-affected and non-affected area. Evolution of the sea surface reflectance, SSR, as an indicator of the degree of volcanic emissions over the sea surface. In situ, bars, and remote sensing, dots. Chlorophyll concentration as a proxy of phytoplankton biomass. Mean volume backscattering strength at 200 kHz as a proxy of mesozooplankton biomass. Mesozooplankton abundance, bars, and dry weight dots from WP2 net halls. 38 kHz representing the acoustic density of the MSL, proxy of migrant micronectin biomass. Table 1. Average micronectin abundance within the MSL during the last survey. Table 2. Average mesozooplankton abundance along the sampling period.